First, of course, I would like to thank you, Stefan, for accepting our invitation to join the TropiBEF workshop 2020 and talk about this amazing project, which is the BEF-China. Thank you very much again. Dr. Stefan Trogisch obtained his PhD from the Institute of Agricultural Sciences of the ETH or ETH Zurich in Switzerland in 2012. He has been working with biodiversity and ecosystem functioning since 2008 when he joined the BEF-China diversity experiment to study nitrogen cycling and the composition dynamics in forests. Currently, Stefan Trogisch is the scientific coordinator of the International Research Training Group, TreeDi, which aims at understanding the role of tree-tree interactions for forest ecosystem functioning. More specifically, they want to elucidate how processes at the individual tree level contribute to the observed biodiversity effects at the community level. Welcome, Dr. Stefan Trogisch, the stage is yours.
Thank you very much, Leonardo, for the nice introduction and for organizing the workshop. And big thank you to your team. And today I'm talking about BEF-China. And let me see if I can move the slides. Yes, it's working. All right, and in the next 30 minutes or so, I will focus mainly on the design of the experiment, and I will give you some information about the establishment and the establishment success of the experiment. If you have time, I will just mention briefly some key results we have achieved during the last 10 years, and I also will outline some future directions. Okay, but first of all, let me start with this map that shows you the global forest area. And as you can see, there are 5 countries in the world that contribute more than half of the forest area in the world. That is Russia, that is Brazil, Canada, United States, and China. So China contributes about 220 million hectare of forest area, which is about 5% of the global forest area. And China is the only country also in the world that has a tropical forest here in the very south on Hainan Island. It has subtropical forest, temperate forest, and at the border between China and Russia, there's even a small fraction of boreal forest in China. So that makes China also a kind of biodiversity hotspot. So in China, there are about 30,000 vascular plant species, and roughly 1/3 of these species are woody plants. So that can be trees, that can be shrubs, woody vines, bamboos. And in China, there are roughly 3,000 tree species and twice as much shrub species. So it's kind of mega diverse country. And I just want to briefly introduce the main forest type that our experiment is addressing. That's the evergreen broadleaf forest. And this forest type is dominated by a subtropical monsoon climate. So you have mean annual precipitation between 900 and 2,000 millimeter per year, and the mean temperature is between 15 and 20 degrees. And formerly, this vegetation type covered about 25% of the country, but it's now heavily reduced to only some remnants. So here you can also see a picture of the Gutianshan National Nature Reserve, which gives you an impression how the forest looks like. And it appears evergreen, but there's also a high fraction of deciduous tree species. Here's some typical tree species or genera, for example, Shima superba, Litocarpus glaber, Castanopsis airi, and Daphne-Phyllum oldhamii. And these trees have kind of single leaves, so it's a very common feature, a glossy leaf surface. So, and many trees having this feature, and so they resemble typical laurophilous leaves. And what is also remarkable is that the forest area in China is increasing. It has increased from 8.6% in 1949 to 23% in 2018. And China is actually leading the list of countries with an annual net increase in forest area. And every year, the forest area in China is increasing by roughly 1%. And this is mainly due to large-scale afforestation programs in China. For example, the 3 North Shadow Belt Development Program, also called the Great Green Wall. China invests heavily in the protection of natural forest, and another afforestation program would, for example, the Grain for Green program that addresses the prevention of flooding and soil erosion by planting trees. So that makes China quite interesting for the establishment of a big tree diversity experiment. So one reason is that it's a hotspot of woody plant diversity. There's a high application potential to integrate species diversity in afforestation schemes. And another important reason is that East China was at that time underrepresented in the TreeDi network. So that's a short history about our experiment. It all started with a workshop. So I think it was in 2004, there was a joint workshop between Germany and China and some international researchers. And there was an idea born, and this idea was further developed over the next years. And there were some field excursions to explore potential field sites. And this initiative resulted in a joint proposal for a DFG research unit. And this got funded in 2008. And over the coming 8 years, the experiment was established. We had 3 cohorts of PhD students working on the platform, and the official end of the research unit was in 2017. And the platform was handed over afterwards to the Chinese side, and the platform, the experiment, is now managed by the Institute of Botany of the Chinese Academy of Sciences and Gutianshan University. All right, so the objectives of the experiment was to answer some fundamental questions. So one question was, what are the effects of tree and shrub species richness on ecosystem functioning, and a large spectrum of ecosystem functions was considered, not only productivity, but also carbon storage, nutrient cycling, prevention of soil erosion, and many more. And we were also interested in the underlying mechanisms. And for that, you have to also quantify the interactions with other trophic levels and strata, for example, interactions between trees and the herb layer, the soil macrofauna, herbivores, decomposers, pathogens, and soil microorganisms. So that's the location of our experiment. Actually, we have the experimental study sites here located in Jiangxi province, about 400 kilometers west of Shanghai. And we have also natural forests where we established plots, and these plots are located in the Gutianshan National Nature Reserve, located here in Zhejiang province. Just a few words to the comparative study approach. Here we established plots, 27 plots, along gradients, crossed gradients of species richness and stand age. And actually, that was the topic for the first cohort of PhD students in the project. So the first PhD students started in 2008. So there were no trees planted in the main experiment. And we had these natural forests as topic for the PhD students. And as mentioned already yesterday, it's also important to have such natural forests in the surrounding, which serves later as comparison to the experimental results. But now to our main experiment. So as already mentioned, we have two sites. We have site A and site B, and here just some key numbers. So the most important thing is that we have very wide gradient in species richness. So we have plots with no tree species and plots up to 24 tree species. Yeah, site A comprises 80.4 hectare, has 271 study plots, more than 100,000 trees were planted, and an additional 44,000 shrubs. Site B is similar extent with 20 hectare, roughly 300 study plots, also more than 100,000 trees and 44,000 shrubs. And now I want to introduce a little bit the design to you. So we used a pool of species that consisted in total of 40 native broadleaf species, and we planted 24 species on site A and 24 species on site B, and there was an overlap in species of 8 species. Then we had different extinction scenarios. We had 3 random extinction scenarios using 3 species pools, partly overlapping species pools, composed of 16 species. And at each site, we had also 2 non-random scenarios or trait-informed scenarios. So I will provide some details on that in a few seconds. So first, the random extinction scenarios. Here you can see all the 40 native species we used for the experiment and the different species pools. And for the random extinction scenarios, we had the 3 different species pools, partly overlapping. And the first scenario of the random extinction was replicated on bigger plots, so-called 4 mu plots. I will explain you in a minute the exact size of these plots. And we replicated this again on smaller plots. And the second and third scenario was only replicated on the smaller plots. For the non-random extinction scenarios, we selected as first scenario specific leaf area. And the reasoning behind that is that species with a higher specific leaf area are going to extinct first. So this is also reflected by natural succession. Or for example, as a drought, then theory predicts that species with a higher SLA go extinct first. And then we had a second scenario that was based on local rarity. And here's the idea that the rare species are going extinct first. So we use these criteria for stepwise elimination of rare species or species with high specific leaf area down to 16, 8, 4, and 2 species mixtures. We also included in our experiment shrub diversity treatment. So there was recently a nice paper by Trias Landjut, and also was emphasized the importance of the understory for ecosystem functioning. For example, they showed that the understory can contribute up to 40% of productivity to the forest ends. And they conclude that the understory communities constitute an important functional component of temperate forests and should not be ignored when developing management strategies to safeguard temperate forest functioning. And we assume that in subtropical forest, with this rich diversity of shrub species, the understory contribution to ecosystem functioning is even larger. And that's why we also included this shrub diversity treatment to our plots. Here you can see a map of the experimental site A with the different shrub diversity levels. And the shrubs were planted on these bigger plots of the first replicate of the first random extinction scenario. So this is such a for a mu plot, we call it, and one mu is Chinese traditional unit for area, and one mu plot is roughly 26 by 26 meter. And then we had per site 35 bigger plots, so-called super plots, where we established the shrub diversity treatment. So we planted no shrubs here, 2 shrub species on that plot, and so on. And in total, we used 18 shrub species, and we planted 10 shrub species per site with an overlap of 2 species. Here you can see the shrub species, and we had also the monocultures of the shrubs, which we planted on smaller plots. We also integrated plots where we planted commercial plantation species. So these were Cuninghamia lanceolata and Pinus masoniana. And these commercial species were only planted as monoculture on 5 plots per site. And last but not least, we also implemented a genetic diversity treatment. So it's of course well known that the species differing in their trait values. So here are, for example, the 4 species: Anophyllum fortunii, Cinnamon camphora, Daphniphyllum ultamii, and Edesia polycarpa. So the species differ, but also the trees of a species belonging to different seed families differ considerably in the trait values. And this can also influence ecosystem functioning. So here are the different seed families for the species, and seed family just means that the seeds were collected from mother trees, and then the seeds were separated and raised separately and then planted in the field. So we know exactly the trees to which seed families they are belonging. And this just the sign, so it's a little bit complicated, but actually we had monocultures of the species here, like in this plot. So we planted monocultures on, so we split it again, the plots, and had quarter plots, and then we established the monocultures. And either the monoculture established only from this 1 seed family, or the monoculture established with 4 seed families. And then we had the 4 species mixtures with the trees belonging only to 1 seed family per species, or we had the 4 species mixtures, but with 4 seed families per species. So this is actually the main design of BEF-China, and this is just an overview here over all extinction scenarios, plot size, number of trees planted. So it's quite complex, but with that design, we can also answer a lot of questions. And now I just want to address some general topics that you probably also will consider when planning your experiment. So the first thing is plot size. So the right selection of your plot size. And plot size depends on the total available area. And of course, the aim of your experiment. So what ecosystem functions you are interested in. And there are advantages for large plots. For example, large plots, they have less stronger edge effects, or between treatment edge effects when the plots are not separated from each other. And in large plot, a more typical and forest-specific microclimate, a more stable microclimate can develop. And probably also larger plots are better for studying multi-trophic interactions if you consider, for example, more mobile organisms like insects, ants, bees, or even birds. Smaller plots or smaller plot size has the advantage that you can increase the number of replicates, and this increase the statistical power to detect plot level biodiversity effects. And smaller plots can also have a lower within plot heterogeneity, but probably also a higher between plot heterogeneity. And one decisive factor is also the duration of the experiment. And the idea behind is that plots should be also large enough to accommodate major trees of each species, also in the high diversity plots. For example, we have plots with 16 species or even 24 species. And here on the left, you can see such a plot planted with 400 tree individuals. But if you then calculate 80 years of forest development, and we know from our observational forests that in average the trees have a canopy projection area of roughly 36 square meters, it will become quite dense in your plots to harbor all the species which you have planted at an early stage. And one solution to that could be to spatially aggregate your plots with the same diversity treatment. So here you can see such a superplot, the 4 mu plots. We have also used for the Shrub-diversity treatment, and the small squares here indicate the trees we have planted, and here the circles are the trees after maybe 70 or 80 years. And if you aggregate smaller plots to such bigger plots, this will also ensure that there's a higher population size of major trees. So otherwise you would probably only have one individual per species, but here if you aggregate these plots, you have maybe 4 individuals per species after maybe 50 years. And then there's the issue about planting density. So we decided to plant trees in a planting distance of 1.29 meter in horizontal projection. So this is about 400 seedlings per plot, or 6,000 seedlings per hectare. And the planting density mimic the natural regenerating forests. It also allows early plant-plant interactions and also rapid canopy closure. So after 2 or 3 years, we had already plant-plant interactions. And another advantage of maybe a denser planting is that this can suppress weeds very easily, because we also experienced high weed growth on the plots, and this costs also a lot of management efforts and some money. Now there's a topic, planting scheme and mixing pattern, and we decided to assign the tree species to, as we assigned them randomly to the individual planting positions on a regular quadratic grid design. And the advantage of this planting scheme is that the local neighborhood can vary, and there's also the fast development of interspecific interactions compared, for example, to patchwise planting or aggregated planting. And this random planting scheme is less error prone than patch planting. So again, we decided for regular planting and then assigned the different species randomly to the different planting locations. And on the next slides, I will briefly give you some impressions how the sites were established. So everything started with the seed collections, with the help of many local farmers who collected a lot of seeds, and we also helped them to collect the seeds, and then the seedlings were transferred to tree nurseries and then raised over the coming 1 or 2 years. So here you can see a picture of the tree nursery with some shading nets to prevent overheating of the seedlings, and evergreen species were sown directly in such pots, and the deciduous species were sown in prepared seedling beds. And for the sites, so here you can see pictures of the sites before site preparation, actually. So that's the status where we have rented the sites. So, and the sites were a former plantation of Cunningamia and pine trees, and that were recently harvested, maybe 2 or 3 years ago. And here you can see some secondary regrowth, and all this had to be cleared before planting, which was actually a huge effort. So we also needed a lot of support and a lot of local workers to get a site prepared. And our former coordinator, Xufei Wang, she coordinated all the planting and site preparation. So she deserves full recognition for her work, and she did tremendously efforts. And then the seedlings were transferred in 2009 and 2010 to the sites. We established some seedling camps, and then the trees were planted again with a lot of local workers on the sites in 2009. So there's a group picture again. So you can see it looks nearly like a desert, and here we employed many Chinese workers that contributed to the success of the experiment. So this was now after planting in November 2009, and a couple of years later it looked green again. So now it looks really nice, the sites. It also feels like a real forest if you're standing inside. And these are some pictures from 2018 and 2019. And you can see that there's a close canopy layer now, and the trees are now between 5 and 15 meters in height. You need ladders, and there are lot of tools to get leaves from the highest canopy layers. just some information about the establishment success. So this was also quantified by Xufei Yang. And after the first planting, the survival rate was just 57%. So some species survived very well in the plots, but others nearly failed completely. And also we experienced that the citrus trees had a higher survival rate and the evergreen species had a quite low survival rate. So we had to replant actually a lot, and after 2 replantings in November 2009 and in March 2010 here, the survival rate increased up to 87% across all species. And what is also important, the right timing of the experiment. And we experienced that the later the seedlings were planted in the year, the lower was the survival rate of the species. And this is to the fact that early spring, so we planted in March and April, and then in April, it's getting really, really hot already in subtropical China. So this was not very good for the survival. And Xufei also put together a lot of advice what to consider when to establish these kind of tree polycultures. So I just refer to this paper here. And we also did a cost calculation, how much the site costs compared to conventional planting. So after 3 years, the height of the trees were between 52 and 300 centimeter. So you can see the different species on site A and the different height grows for the height or the crown diameter or the canopy width. And we also noticed that the plot level mean growth was mainly driven by community-weighted mean trait values and the functional diversity, but less by the environment. So that was quite surprising because we have also quite complex topography. And here are just some key results I want to present. So after 8 years of growth, we found that the highest tree species mixtures stored twice as much carbon as the average monoculture. So, and we could also show here in this paper that the trend or the, how to say, the relationship gets steeper and steeper from year to year. Maybe some other results. So we have seen that tree species diversity can promote also community wood productivity and carbon storage. And now we are looking more into the mechanisms, for example. So Andreas Fichtner et al. Found, for example, that the neighborhood interactions mainly drive this relationship. So they used neighborhood interactions as predictor of community wood productivity and found that neighborhood interactions really explain a lot for the community wood productivity. And also Matthias Kunz used terrestrial laser scanning and found that also the crown complementarity increases with neighborhood tree species richness. But tree species richness is not everything. So Mingcheng Wang was here interested in the different multi-trophic interactions, and he found, or first he collected more than 8,000 butterfly larvae for more than 25,000 trees, and he found that the phylogenetic tree species composition actually relates much better to the species composition of the herbivores here or the caterpillars. So this has also some implications for forest restoration. So maybe you should also select the right tree species composition to restore the species composition of higher trophic levels. And I want to close with 2 slides about our new international research training group, TreeDi. So this has started in 2018. So it's a bigger project as well, and it involves several universities on the German side and on the Chinese side here. And what is unique is that all our PhD students doing work on the BEF-China platform in complementary projects. So for example, on the German side here, the first project focused on above ground complementarity and productivity, and on the Chinese side on below ground complementarity. And the topic is to look more closely on the individual neighborhood interactions and the immediate or the local neighborhood. So we established so-called tree species pairs in the different local neighborhoods and plot tree species richness. And the goal is, or the intention is, to quantify the effect of the local neighborhood interactions for community-related processes. And the platform is also open for collaboration. So this is the current steering committee of the platform. Professor Kipping Ma, Helge Poolheide, Bernhard Schmid, Professor Yanfeng Wang. We have the president of Gutianshan University and Xiaochen Liu. Okay, and I want to close with a big thank you to all the people involved in the experiment, to all the funding agencies, to all the former PhD students, and I would like to thank you for your attention. Thank you.